I am Dr. Waseem Sajjad, and from today onwards, we are going to start neuroradiology, the imaging modalities which are used during neuroradiology. And in the first lecture, we will discuss about brain tumors, the imaging modalities used for brain tumors, and classification of brain tumors. So the most important imaging modalities of neuroradiology is the CT scan brain and MRI brain. These two imaging modalities are the hallmark of neuroradiology, and there can be contrast enhanced CT and different sequences of MRI which are used to image the tumors and other pathologies inside the brain. So, if we talk specifically about brain tumors, the CT scan is used as the first imaging modality when we are suspecting brain tumors. The non contrast enhanced CT scan is done, and the brain tumors mostly appear hypodense on non contrast enhanced CT scan, but the tumors which have calcifications are if intratumoral hemorrhage, then this appears as hyperdense. Contrast enhanced CT scan also provides valuable information about the brain tumors, integrity of the blood brain barrier, and the enhancement of the brain tumors. The more the enhancement, the more aggressive the tumor is, and the more disruption it has caused to the blood brain barrier. Now, a recent imaging modality which is used for brain tumors is the CT perfusion. CT perfusion detects the relative cerebral blood volume inside the tumor. It basically the measure of tumor vascularity, and it is different from the MR perfusion imaging because in CT perfusion imaging the area which we can image is relatively smaller, and the benefit is that we can measure the density of the contrast, how much dense the contrast enhancement is in the tumor, and we can also compare the densities of the tumor and the normal brain parenchyma. So, non contrast enhanced CT, contrast enhanced CT, and CT perfusion imaging. These are the three imaging modalities depending upon the CT scan. And now, the most important imaging modality for brain pathologies is the MRI brain. Different sequences of MRI brain are used to image the brain tumors: T1 sequence, T2 sequence, T2 flare. T2 flare is the most important sequence in which the tumors can be distinguished from the brain parenchyma. It provides a very good contrast when tumor and normal brain parenchyma is compared, and T2 flare basically measures the fluid quantity inside the tumor. The more cellular tumors like high grade lymphomas are less hyperintense on T2 flare sequence as compared with those tumors which have high water content. After T2 flare sequence, T1 weighted images are important. T1 weighted images are important for calcifications, for hemorrhage, for fat, for melanin. If the brain tumors have hemorrhage, then they will appear hyperintense on T1 images. If the brain tumors have calcifications, they appear hyperintense on T1 weighted images. If there is melanin inside the brain tumors like metastasis from melanin forming tumors, then it appears hyperintense on T1 weighted images, and if there is fat inside the tumor it also appears T1 hyperintense. And then there are T2 star sequence. T2 star sequence is specifically important for hemorrhage and calcifications. Hemorrhage and calcifications are more conspicuous on T2 star images as compared with other sequences. Now we discussed T1 weighted, T2 flare, and T2 star. Now the next important sequence is the T2 susceptibility weighted contrast enhanced images. This sequence measures the relative cerebral blood volume, and This sequence is also called MR perfusion imaging. It is similar to the CT perfusion images, but the MR perfusion images are more have more contrast when compared brain parenchyma and the tumor, and MR perfusion images can image a wider area as compared with the CT perfusion images. MR perfusion images is a direct measure of tumor neovascularity. When there is high signals on MR perfusion imaging, it means that the relative cerebral blood volume in that specific area is high. And when relative cerebral blood volume in a tumor is high, it means the tumor has neovascularity, and neovascularity means the grade of the tumor is high. So MR perfusion imaging helps us to grade the brain tumor. The more intense signals on MR perfusion imaging means that the tumor is of high grade. Similarly, there is dynamic contrast enhanced images. Dynamic contrast enhanced images are different from the MR perfusion images. Dynamic contrast enhanced images basically measures the integrity of the blood brain barrier. If the blood brain barrier is disturbed, then there will be enhancement of the tumor and it appears hyperintense on contrast enhanced MR images. For contrast enhanced MR images, we should compare the same sequence in non-contrast enhanced MR and contrast enhanced MR. Suppose if we are imaging T1 sequence on contrast enhanced images, we need to compare contrast enhanced T1 weighted images with non-contrast enhanced T1 weighted images. We can't compare contrast enhanced T1 weighted images with non-contrast enhanced T2 weighted images. So the same sequence is used and then contrast is given to measure the contrast enhancement, and it tells us about the integrity of the blood brain barrier. So there are few latest techniques like arterial labeling of the hydrogen atoms. This arterial labeling of the hydrogen atoms tells us about the cerebral blood flow. The more cerebral blood flow to the tumor in arterial hydrogen labeled arterial MR sequences, it means that the cerebral blood flow to the tumor is high. It is different from the cerebral blood volume. Cerebral blood volume measures the vessel density inside the tumor and it is a measure of tumor neovascularity. So the next sequence which can be used for brain tumors is the MR diffusion images. MR diffusion is a very useful sequence. It basically measures the ability of hydrogen atoms freely move inside a specific space. So if the tumor is highly cellular, this diffusion will be restricted and we get a high signal on diffusion weighted images. Diffusion weighted images are always compared with the ADC map. ADC map is the apparent diffusion coefficient. If there are high signals on the diffusion weighted images, there should be low signals on ADC map, because ADC map is the true measure of the diffusion of hydrogen atoms inside a specific space. So diffusion imaging, the latest technique which appears in diffusion imaging is the diffusion tensor imaging. With the help of diffusion tensor imaging, we can also measure the direction of movement of the hydrogen atoms, and it is very important when we are measuring the white matter tracts and we are imaging the white matter tracts. The different white matter tracts has different direction of diffusion of hydrogen atoms, and this direction of diffusion of hydrogen atoms is disturbed if white matter tracts are affected by the tumor. So diffusion tensor imaging is the measure of an isotropy of the white matter tracts, and it is useful to know whether the white matter tracts are affected by the tumor or not. Now the next sequence is the MR spectroscopy. MR spectroscopy is a very sensitive imaging sequence, but its specificity is relatively low. In this sequence, we place an area of interest on the tumor, and the MR shows us the different concentration of different ions inside that area of interest. We measure the concentration of creatine, we measure the concentration of N-acetyl aspartate, we measure the concentration of choline, lactate, and lipids, etc. Now, N-acetyl aspartate is the representative of normal neurons because it is present in the normal neuronal tissue. If N-acetyl aspartate concentration inside a brain area has decreased, it means the neuronal tissues inside that area has been affected and the normality of the neurons has been disturbed. So in brain tumors, N-acetyl aspartate will be low. Similarly, creatine peaks will be low, but choline and lactate peaks will be high. Choline is the measure of cell membrane turnover. In a tumor, there are a lot of cells which are dividing at a rapid pace without any inhibition, and there is a very rapid cell membrane turnover, and that's why the concentration of choline is increased when there is a tumor. Similarly, lactate is the measure of anaerobic respiration of the cells. When there is a tumor, the blood supply will be compromised. Lactate appears inside the brain tissue and it appears as high peaks on the MRS images. In addition to these images, there is functional MRI images which have developed recently. In functional MRI images, we can measure the amount of blood flow inside a specific area of the brain. When we are thinking about a certain thing, the concentration of blood increases in that area of the brain which is controlling that specific thinking. So, in functional MRI images, we can even tell what the patient is thinking and what are the areas of the brain which are active during the scan time. So this is a very useful MR sequence specifically used to know the functionality of brain parenchyma. The tumor or some other pathologies affect a certain area of the brain, then the functional MRI helps us whether this area of the brain is functioning normally or not. So these are the basic MR sequences which are used in tumor imaging in addition to the CT scan images. In the next lecture, we will discuss the different types of the tumors, how we classify brain tumors, and how on imaging different types of the tumors appear, and how confidently we can tell the tumor is intraaxial or extraaxial. Thank you.